So let's get started. In this talk, I'll show examples how one can implement acoustic dispensing into high-throughput single-cell genomics or single-cell sequencing workflows. But at the beginning, let's remind ourselves, why is it worth examining single cells and not just do bulk sequencing or bulk analysis like we used to do before? Well, if you do this, so let's say you have this button, a population of cells, isolate them, isolate RNA, do some gene expression analysis, you end up getting some numbers for your genes, 1, 2, and 3, expressed in those cells. Now, if you're able to dissolve this population and look at each cell individually, you might see a very different picture from what a bulk information gives you. So by looking at individual cells, for example, here you can see that we have at least 2 populations in here. We have the ones that express blue and yellow, and one that expresses green but hardly any blue. So this might be a transition between 2 states of cells or 2 distinct cell populations, which you would not have seen just looking at this type of data. There's also benefits when it comes to single-cell genome sequencing. Let's say these are your read piles. You see 2 somatic variants, a red one and a green one, and yeah, okay, what does it mean? Are these variants present in the same cell? Are they present in different cells? Are these maybe tumor subclones? So with single-cell sequencing, you can start to actually address these questions which bulk methods just wouldn't have given before, so that our understanding develops from blue dots to colorful dots with different properties in our tissues of interest. Current workflows have 2 big parts to it. The first one is isolating single cells, which is still a huge issue, and many people started working on that. The second one then is how to amplify and create some material that we can feed onto our high-throughput sequencing machines. For isolation from single cells, there's various approaches possible. The most direct one is probably laser capture from just tissues into some form of reaction vessel. Most people at the moment use dissociation approaches to get single-cell suspensions, and then there's a range of methods, most commonly used cell sorting. There's also ways to use micromanipulation or microfluidic machines like the devices from Fluidigm or droplet platforms that in the end all yield a cell in some form of reaction container. A typical human cell contains 6.2 picograms of DNA and typically less than 50 picograms of RNA, which is why we need to amplify material. Typically, you would then have to make a decision whether to go down the DNA route and use some either MDA or PCR-based methods. There's now also, well, it was published last year by our collaborators, single-cell bisulfite sequencing, or you dedicate yourselves to RNA transcriptome sequencing. There was a paper 1.5 years ago by Ian McCauley. He's now at, not TGAC, the Earlham Institute. So it's called GNT-seq, which actually allows you to do both things on the same cell by separating DNA and RNA. But the things I'm going to talk about here still apply to this method, as this is a physical separation of material. In this talk, I'm going to focus on the RNA sequencing aspect of single-cell genomics and library preparation protocols. And in the beginning, I just want to showcase one experiment that demonstrates the power of single-cell RNA sequencing. So this was done by Ola Kolodzic in Sarah Teigman's lab at the Amplification and Sanger Institute. She's now moving to Weizmann, so if you want to get in touch with her, you need to go to Israel. Her experiment, what she did was she took male C57 black 6 mice and this other strand with this long number, female, crossed them, and from the blastocyst isolated the inner cell mass and seeded them into different dishes, so basically have stem cells, and treated them with different media. So one is, or 2 media are with GSK3 and MEK1/2 inhibitors, which are deemed to keep the cells in a ground pluripotent state. And then there's a treatment with serum and an inhibitory factor, which is thought to transition the cells into a more differentiation permissive state. She then applied single-cell RNA sequencing on these cell populations, as well as just naturally developing blastocysts. And this is a principal component analysis. So these are the three main contributing factors to the variation in these cells. I think the total data set here is about 2,500 cells. And what we see here are many zygotes, so true single cells, and that's something you can only look at really with single cell sequencing. We then see the developmental trajectory to the inner cell mass, and then three different populations, which is a ground state, an intermediary state, and the differentiation permissive state. And what you can already see is that the heterogeneity in these different populations increases as cells become more differentiation permissive. So basically make decisions along that developmental tree where to go next, so to say. Some deeper analysis then showed her that these populations, so there's these differentiation path cells that are committed, and then there's these other two populations, the differentiation primed cells and the ground state cells, and they seem to be cycling. So there are cells basically going to this state, maybe differentiate, but maybe not, and go back. And this analysis is based on the analysis of the distribution of gene expression across the population. So just one example, OCT4, the red population, the cells on the differentiation path, you can clearly see that there are two distinct populations of single cells, some that express a high level and the majority actually expressing a low level. If you just had an average readout of this, you would probably get a single data point about there, which would not be too different from the other two populations. So single cell transcriptome sequencing in that case just increases the resolution and allows you to see cycling of cells between different states. If you remember a slide ago, she used two different mouse strains, which leads to the situation that most genes are heterozygous or express heterozygous SNPs. So by just calling a known SNPs in the transcriptome, she could then detect whether a transcript was expressed from the paternal or maternal allele and whether there's differences between genes or different states in the cells. Just as an example, one gene that's equally expressed from the paternal and maternal allele, one gene that's exclusively expressed from the maternal allele, another one just exclusively paternal. Just to showcase what types of experiments are possible with single-cell RNA sequencing. Our workflow consists of four major parts. The first one being cell isolation, which I am not going to cover today, other than we use a BD influx to sort cells into 96 and straightforward plates. And I could probably give another 1 hour talk just about this issue, which I'm not going to do, considering the whiskey. So our workflow in our lab actually starts with reverse transcription of single-cell RNA and amplification. We then have a cleanup step and then a library construction pipeline to convert cDNA into sequencing-ready libraries. The protocol we use is SmartSeq2, developed by Simone Pichelli at the Karolinska Institute and SciLife Lab in Stockholm. And this protocol works as such that you take your transcript, you do a reverse transcription with an oligo-DT primer that contains an adapter sequence. You initiate reverse transcription, so you get your strand synthesis, and MMLV-derived reverse transcriptases append these three non-templated bases, which can then be used to prime off these three bases with just an LNA base here in the middle to increase the melting temperature of this little thing. Then what happens is that the RT enzyme extends your transcript and incorporates the second adapter sequence into the first strand. So this is called template switching. And now we have a transcript with a universal handle on both sides, which we can just amplify using PCR. In practical wet lab terms, this protocol has three pretty simple steps. There's a lysis buffer, so you need to put your cell into a lysis buffer to lyse it. A reverse transcription, and then PCR amplification of the first strand cDNA. These are the published volumes of the protocol, so 4 microliters, 5.7, 15. This is how you would do it with a regular pipette. Of course, being a high throughput facility at a large institute, this is not how we do things anymore. What we needed was some automation platform with 3 key requirements. We wanted rapid few-to-many transfers, so we only have 3 reagents, but many wells on many plates. It needed to be sterile. RNases are a massive concern if you're dealing with 50 picograms of RNA. And it should be easy to use for the lab user. And for the time being, our system that we use is the Mantis, not an Echo. So here you can see for Labcyte Mantis, we've slightly modified ours and built a chilled nest onto it. So this machine, they have molecular biology grade consumables that are actually RNA-free, so we tested those and they are clean, which is not always the case. You can buy, so the way it works, you have this little arm here, and then you have these chip positions, and you can, so there's 2 here and 4 there, and you can see here's a tube running into this big Falcon tube. So master mix goes in here and then through the tube. There's 2 chips, 100 and 500 nanoliter chips, so it has 2 chambers, uses direct displacement to aspirate and dispense droplets, and a large volume chip or high volume chip, 1 microliter or 5 microliter. You can feed 6 reagents into it. A straightforward plate, depending on the volumes that you have, takes between 3 and 9 minutes, which is not blazingly fast, but for the time being good enough, considering we can chill our plate. What we've then done is, since these volumes don't really fit nicely on an instrument that does these small increments, is change these volumes. So for example, for 5.7 microliter dispense, you would, if you wanted to actually dispense 5.7 microliters, do 9 times 500 plus 2 times 100. And then your plate takes 15 minutes. So we've tweaked the protocol to make it fit 1 or 5 microliter dispensers. For the current version we are running in 384 plates, takes 1 microliter lysis, but for 1 microliter RT mix, 5 microliters PCR mix, just a single dispense. We are going to replace this, also not with an Echo, sorry Devin, but a Bionex nanodrop. And the whole workflow that we're then doing in the lab is dispense on the Mantis, seal, shake, spin, and then incubate for 3 to 5 hours, which is the main reason why we haven't gone for any integrated kind of high throughput platform, because that's a massive bottleneck in the whole process. So this part of our process is manual processing. So you can see PCR hood with the Mantis, seal, shake, spin, and then tetrads are about here. So slightly different view on the same process. We provide our customers with pre-filled lysis buffer plates, so 1 microliter on the straightforward plate, 4 in a 96. They go to our sorting facility with their cell suspension. The operators there sort single cells into those plates. They bring them back. We add reagents, do some thermocycling. So 1 tetrad gives you 4 96 or straightforward plates. And before we progress to library preparation, we'd like to have some form of quality control on the cDNA that we generated. So what we're now starting to do as a process improvement is that we take a Hamilton star, straight 4 head, and this robot now samples 24 wells across each of the plates, from top left corner, bottom right corner, and across the middle, and puts this into a 96 well plate. And then this 96 well plate goes into our new toy here, which is a fragment analyzer as a 96 capillary array, and within an hour you get a picture like this, which is now 96 cDNAs. And yeah, basically we can just confirm whether we have sufficient material to continue and what size it is. So this is really valuable to spot plates where we had degradation of the RNA due to storage, contamination, endogenous RNases, or just age of the plate, reagent failures. There's all kinds of things that can go wrong, and this system now allows us to look at per input plate 4 controls and 20 single cells. Before that we were using a bioanalyzer, which allows 11 samples. Just a slightly zoomed in view, so you have this electronic gel, which is basically a representation of these traces, which give you size versus quantity. We have certain pass-fail criteria. The next step for us will be that we then use this quantity to drive our downstream stamping onto Echo source plates, which I'll come to in a minute. So far we've covered, well, not really cell isolation, reverse transcription, and cDNA quality control. The next step in our process is cleanup, which is done on the same Hamilton straight 4 star. It's a conventional pipetting robot, so for anything that's bead cleanups, we're still reliant on these systems. I'm not going to go into too much detail, just one thing here for the validation of this assay. When you look in the literature, how do people validate that their spry bead to DNA ratio is correct? Everybody says, oh, just use a ladder. Well, we've done that, it gives you wrong results. You need a sufficiently complex mixture to really see how your spray cleanup performs. So we've made a mix of a DNA letter, which you can see here, and then two different materials, so genomic DNA sheared to 250 base pair and 1.5 kb, and spiked the letter into that. So we now have a complex mixture. We put it into right buffer and then perform the cleanup, and this is basically just a validation showing non-cleaned material in green, manual in red, and the robot in blue, so it works fine. A single plate takes 45 minutes. We're now working on a parallelized version that will hopefully do 4 plates in 90 minutes. And the output of this process is cDNA in an echo source plate. Here we are. So we had material, sample amplification, sprite cleanup, and then we have an echo source plate. Now, the next step for us before we go into library preparation is that we want to quantify the cDNA content or genomic DNA content of every sample that we feed into this. And this is now where the echo comes in place, which is the central instrument in our library construction pipeline. So most of you probably have seen one. So we have an Echo 525 integrated via the Access system with a CombiNL bulk dispenser, a Fluostar plate reader, a plate lock, a centrifuge, a D-litter, and an on-deck thermocycler for straightforward plates. And the next step now I'm going to talk about is how we read and normalize samples on this system. And I still remember that experiment that I've done with John on our demo echo back then. At that point in time, we didn't really know whether it could transfer DNA sufficiently well. So this was actually the key experiment for us, where we took 3 different types of DNA, did a manual transfer, 1 microliter with a pipette from a tube into a quantification plate, and asked the echo to do the same thing. Transfer 1 microliter of a known DNA sample. So these are dilution series, twofold, and if you get a perfect transfer, every dot should sit along these lines. The materials were tested were genomic DNA, 1.5 kb, which transfers fine up to 100 nanograms per microliter, 250 base pairs, sitting right behind here. Lambda DNA, which has a length of 47 kilobase pairs, transfers fine up to a point of 25 nanograms per microliter. Genomic DNA, this was Promega genomic DNA, average size 130 kilobase pairs. We started to see this very interesting effect. So we can transfer this manually, but the echo at one point stops transferring genomic DNA. This is about 3 to 5 nanograms. So this is for very long fragments, obviously. We then looked into this with Labcyte. So I don't know, do you share that with you, that video? So the reason is you get long fragments that align with each other, and basically the droplets, as they eject, are pulled back like on a bungee jumping rope. For us, fortunately, that's not an issue. Our cDNA is at maximum 5 kilobase pairs, so we can transfer up to 100 nanograms per microliter, which are quantities we never generate. Another very encouraging thing about the echo, so this is just looking at the CVs of these replicates. So blue is the echo, so we get about 1% to maybe maximum 4% CV. Yellow, that's me pipetting. I think I don't need to comment this.
No whiskey.
No whiskey. Maybe more whiskey. Again, we had to validate this, the DNA quantification, and the aim of our DNA quantification process is to quantify 8 straightforward plates in one run. Right? It's high throughput, so we were looking at many samples here. The key questions were, how can we fit standards into this process? And is our normalization efficient that we're doing? So, I mean, straightforward thing is you take sample plates 1 to 8 and just do 1-to-1 stamp onto your quantification plate. That's fine, so you get your 8 times 384 readouts of your samples, but what about the standards, right? You can take a standards plate, put just standards on and read it, but then your standards might, what we've seen is that between individual plates there's variations in backgrounds, there's slight shifts. So what we came up with is an approach where we create, using the echo, a pool of all 384 samples on sample plate 1 in 6 replicates onto the sample plate, and do this for each plate. So for each input plate, we create a pool of all 384 samples and read that pool alongside the standards. And then there's some math happening in the background. So we first measure the standards on the plate. We then measure the pooled samples, so the real pools, and calculate the concentration against the standards. We then measure all 8 plates of our individual samples, again calculate the concentration. And then we calculate the average sample concentration on the sample plates. And in theory, if you have no drift between your plates, that should match, the calculated standard should match the actually pooled sample standard. That's never the case, so there's always slight differences. And then all that we do then is basically correct our samples for the relative difference between the calculated pool concentration and the measured pool concentration using this formula. So you take your measured, so your real sample concentration is your measured sample concentration plus the difference of your measured pool and your calculated pool weighted by the measured concentration multiplied by the measured concentration divided by the average of all samples. And then this means, let's say your real sample is 10. No, sorry, what was I say? The difference between your measured pool and the calculated pool, let's say 1, so you would correct a sample that's 10 by -1, whereas a sample that's measured as 1 by 0.1, so it's basically you just shift a linear curve. Does it work? So to address this question, we took a cDNA, so real-life material, at 35 nanograms, spiked in some amplicons that we could easily detect by qPCR, then did a serial dilution, which means your cDNA gets diluted and your amplicon gets diluted at the same rate, and then did the qPCR. So, and the theory is, if the normalization works, we should just see a flat line for our samples. What we can see here is that between, so these are the dots, so this is the concentration of the cDNA, this is just an arbitrary qPCR value. What we can see between 2 nanograms and 20 nanograms, we get a normalization to our defined target, which is now the range we can work in. And yeah, it's good enough for us. Of course, here you can see at one point, if your sample becomes too dilute, all we can transfer is 25 nanoliters or nothing. So, and on the other end, the maximum, sorry, that applies here. On the other hand, we have a maximum volume of 500 nanoliters of sample that we want to use in the downstream process. So at some point, we're limited in terms of how much sample volume we can put into a single well. So after the quantification now was out of the way, we went ahead and developed a library preparation process. We decided to go with the Nextara process to make libraries. This is basically taking your DNA and transposase, which cuts your fragments your DNA, inserts a small overhang, and then you can run a PCR with an adapter and an autoprimer against these overhangs to introduce sample barcodes here and here and flow cell adapters. It's a very nice protocol. It's really fast. It only takes 2 hours to perform, and just using a regular pipette. And there's 2 kits out there that you can buy. So there's Nextara XT kit, which is marketed for small genomes. It takes an input of 0.2 nanograms per microliter, 5 microliters, so 1 nanogram of DNA. Add some segmentation buffer, you incubate it for a while, you neutralize with the buffer, and then do this PCR. There's another kit, which is the Nextara Genomic DNA kit, which uses 50 nanograms of DNA, so actually quite a lot, does a high volume reaction of fragmentation. Then this protocol asks for a column cleanup to remove the transposase, which is tightly bound to the end of the transcripts, and then does a PCR. Now, when we looked at this protocol, we liked the fact that it's 2 hours. 50 nanograms input of the Nextara is really too much for us because we would have to amplify a lot more material from our single cells. The 1 nanogram Nextara XT is already pretty good. That's also one of the recommended protocols to use with the Fluid IMC1 system. Big challenges, if you do it manually or on a tip-based instrument, you use 10 tips per sample just due to the indexing and the number of pipettings that you have to perform, and it's really expensive. It's £50 per sample, 24 for the cheap one, which obviously is a big inhibitor for large-scale projects. So we went ahead and basically took this protocol, Nextera, and modified it to run on an echo in a miniaturized way. Since the library size depends on the Nextera enzyme to DNA ratio, the accurate quantification is essential. We've demonstrated that. And this protocol works with the dual indexing approach, which means you have a forward index primer and a reverse index primer. So 20 primers encode 96 samples, 40 primers can encode 384 samples. We are now working on the 1536-plex index set using 80 primers. To cut a long story short, this is basically what we ended up with in the end. So our modifications where we reduce the volumes. So we now use 500 nanoliters of DNA normalized to 500 picograms. We only use 100 nanoliter of the enzyme buffer mix, opposed to initially 30 microliters. This modification alone leads, even including the echo dead volume, leads to a situation where instead of 96, we get about 6,000 reactions out of a single kit. We use STS to neutralize. This is basically what the buffer NT of the Nextera XT kit is. It's just STS. It denatures the transposase, so it falls off the ends of the transposons. We use kappa hi-fi as the PCR enzyme, so we replace the Illumina enzyme with kappa hi-fi. We have now our own indexing primers, 125 nanoliters, auto-PCR primers, so total reaction volume of 2,400 nanoliters, opposed to 50 microliters originally. And just one example, this is what a library looks like after performing Nextera XT as the manufacturer prescribes it. This is libraries as they come off the echo in our hands with this protocol. So you can see it's the same performance. This is actually a bit short, so this is still needs some cleanup. So we're aiming for libraries that start at 300 base pair and extend to about 1,000 base pair. So, but this is just then some polishing at the end of the process. This is how the process is done on the Echo Access platform. So there's many steps in here. So initially we take cDNA or genomic DNA, put this onto a straightforward PCR plate, backfilled with water. That's the first step. Then against the same plate, we transfer our Nextera enzyme, incubate for 5 minutes on the on-deck thermocycler, neutralize with STS, again, same plate, incubate for 5 minutes, and then add PCR, primers, master mix, seal the plate and park it. Because what happens next is a 12-cycle PCR, which we are not doing on deck in order to not block the system. So just visually a bit more pleasing. So we have a normalization step, tagmentation, neutralization, and PCR. We then went ahead, and so this is running fine for a single plate. We then went ahead and, of course, wanted to do many plates. And this is where we reached one point where we got a bit stuck. Because what happens is with the current version of the Tempo software is that it takes all plates through step 1, all plates through step 2, step 3, step 4. So just replay, does this, then all of these steps, all of these, all of these. Which is fine in high content screening applications, but we have time dependencies here. At this stage, the sample is 500 nanoliters. We can't wait for another 7 plates to finish normalization while this is sitting there. Then we add the enzyme, it should go in the thermocycler immediately. It should also come off immediately and be neutralized immediately, otherwise our libraries become too short. After that, we're a bit more relaxed, but again, evaporation might be an issue. The PCR is actually fine. So the situation we're now working on with Labcyte, and I don't know if this is part of your Labcyte 2.0 to a 3.0 transition, or if it's 1.9 to 2. What we want is a process that basically runs one plate through all steps before it pulls in the next plate. Right, so we've now started to work on this with Labcyte, so we'll see how this goes. But so far, these guys have been really helpful and approachable, so that hopefully at some point in the future we'll have workflows that allow basically any number of dispenses against a single source plate before it starts to parallelize the next plate. So at the moment, this is unfortunately a process where basically one of my guys is playing access arm, which is not how it should be. After the library preparation, we use a Hamilton star 8-channel system to create pools of 96 or 384 samples into tubes. Then we have a library QC for which we use a bioanalyzer and a Labcyte 384 for qPCR. So just a bit more detail. So we first use a bioanalyzer to estimate or to assess the size of the library and get a quantification estimate. I'm deliberately saying estimate because that's all you get from a bioanalyzer, and people should be aware of that. We then do a pre-normalization to less than 10 picomolar concentration of library, then do a 1 in 1,000 dilution on the robot, and then perform a qPCR. And after that, sequencing. So for the qPCR, just one more thing I want to show here. Initially, we tested the kappa library quantification kit, or that's the one that we use. So in this top chart, what you can see here is the library concentration in picomoles, just using the 6 standards they provide with their kit, which cover quite a wide dynamic range. And on the left-hand side, you can see the quantification cycle as it comes out of the light cycle. The right axis is these dotted lines, these are the CVs. The black curve shows the how the standard curve looks like in a 10 microliter reaction. The blue curve is 5 microliters and the red curve is 2.5 microliters. In case you can't see different colors, that's because they are all on top of each other. Right? So another benefit of the Echo, or another application where we really like it, is qPCR, just miniaturization. We went from a standard assay that's 20 microliters down to 2.5. Our R square is still 0.9987, which is pretty okay for a standard curve. Another thing that the Echo allows us to do is to basically simulate a serial dilution of libraries. The standard process, as kappa describes it, asks you to provide a 1 in 2,000, 4,000, and 8,000 dilution of your library to make sure that at least one of these concentration falls into your standard curve. So normally this would be quite a lot of pipetting. What we do here is basically transfer 1,000 nanoliters of sample, 500, 250, and 125. And this graph shows what the expected molarity of a diluted library is, and this is what we measured, and it's right on top of each other. So the transfers for short fragments are just perfect in that respect. So looking at qPCR, it allows us to skip a dilution series, which can also be kind of error prone if it's done manually. It's also not that straightforward on a liquid handling machine. We can reduce reaction volume to 75%. We have no other consumable cost than the plate, so in total, just on qPCR, we save about 75% of the cost this would normally cost. Then we go for sequencing, so this is an image from our sequencing core facility. I'm not going to cover that. So to summarize, through the 3 main steps in our process. So for isolation of single cells and the throughput we can achieve, that's basically unlimited, maybe limited by staff time, how long someone would want to sit in front of that instrument. For amplification with the current setup using Demantis, just thermocyclers, and this is for GNT, which we haven't covered here, you can easily do 12 straightforward plates a day with the SmartSeq protocol. MDA easily 36 plates, so you can do 3 runs in a day. It's only a 2-hour protocol. Picoplex is a bit longer. You can still fit into runs. You're basically limited by the number of PCR machines that you can fit into your lab and the length of your PCR protocols. When it comes to the library preparation, our spray cleanup, once parallelized, will deliver 20 plates per day. So that's not a limitation in the current scenario. With the Nextera, we're currently limited to 8 straightforward plates a day, just due to the fact that the walkaway is not yet possible. It's just, at the moment, it takes a lot of concentration to get this right. So hopefully at one point we'll have a fully automated version, and then we should be able to hit at least 16 a day. And the real bottleneck at the moment is pooling on the Hamilton robot. It's moving back and forth a lot. It takes over an hour, so realistically we can only pool 6 straightforward plates at the moment. Now, the good news is there's a low dead volume plate coming soon-ish, hopefully, in which case we then might be able to do pooling on the Echo as well. That's it. So that's my team. That's Irad. He's here as well, so he developed most of the library preparation aspects, so any details, ask him. This is Andrew, our bioinformatician, so he's dealing a lot with software integration and the LIMS. These are Joe and Emily, our wet lab staff that actually work and process samples. And yeah, I want to thank these guys for all their effort, Labcyte for inviting me, A, and B, supporting us, and you for your patience. Thank you.